Yeah, good afternoon. I hope that you could replenish your brain with glucose and not so much your guts with blood. We can go ahead with the afternoon session. In my presentation, I will cover a relatively broad range of topics, obviously, spanning from patient-reported outcomes of PROs to molecular or imaging biomarkers and to novel methodologies. And here I will briefly discuss optical coherence tomography, and I'm very pleased that the speaker after me independently selected the same topic. So that's in a way a good thing. All these novel outcome measures, except the established MRI markers, may play an increasingly important role in the future drug development progress in multiple sclerosis, and therefore they may deserve an adequate coverage in this guideline update. How does this work? Like this. So let's talk about patient-reported outcomes. And I use here a definition I found in a CHMP paper of 2005. It is any outcome evaluated directly by the patient himself and based on patient's perception of a disease and its treatment. And you see on the right hand of the slide examples of instruments in that space, and you see it's a broad range which is covered. It goes from health-related quality of life measures over impairment measures to adhesion and treatment satisfaction measures. So it's really broad, and it's not an easy topic by itself, because you really need to think what you are talking about if you deal with them. Some of them have been used in MS, and reliability and validity have been assessed. This is the wrong direction. So why it is important to cover patient-reported outcome in the MS guidelines, and I think that had been already touched upon in the morning discussion. They are important tools to capture patients' perspective, and they complement and support meaningfulness of other measures. So we very often are left with an objective measure which gives you a couple of seconds. We had the discussion this morning on the walking test, but what does it mean? And I think, or we think, and that is a context where patient-reported outcomes may play a major role. The current draft guideline limits its coverage to quality of life, and it gives only relatively unspecific guidance regarding the quality of scales which should be used. It uses a set of general terms, reliable and validated, which of course is true, but what is it exactly, what do we need to do in that context? And we heard already this morning that this is not so easy, but nevertheless important. So several pros have undergone some psychometric testing and have demonstrated some validity, reliability, and responsiveness. And we specifically, and again, it's quite nice to see some convergence this morning. Several times the MSWS12 came up in the discussion, and we also felt that this would be a scale particularly relevant because it has been used in recent regulatory processes to support the clinical meaningfulness of improved walking speed. And that is why that scale probably could be a kind of an anchor point, what we all believe could be useful and sensible to use. I will learn to do that at the end of my talk. The recommendations we have here that the guidelines should probably consider to cover a broader range of patient-reported outcomes beyond quality of life, and probably the MSWS12 could be specifically mentioned as an anchor, as an example of a scale which, at least from its properties, is useful enough, deemed useful enough by regulatory authorities to be useful in a drug development process. And we'd also see the shortcomings in that field, and that is why we also call for collaborative efforts from all sides concerned, industry, academia, and regulators, to develop criteria for appropriate pros, which in turn would then help other people to develop these instruments into the right direction, and thus give more coverage. I will now turn to the next topic, which is biomarkers, and we have split this into imaging biomarkers and cellular or molecular cellular biomarkers, which I cover first. The draft guideline quite rightly encourages the use of biomarker to identify patient subgroups. It acknowledges the importance of a search, and here I think the important word is already there, the search of valid biomarkers. Not so many are around, to improve the efficiency in exploratory trials. And I personally like very much it encouraged to include a biomarker program as an integrated part of a development program. Why is that important because we are still in quite of dire need of appropriate cellular or soluble biomarkers in the MS space for many, many reasons. There are very few which have been raised to clinical utility, such as a GC virus test for risk stratification for natalizumab treatment, or possibly anti-aquaporin for antibodies for differential diagnosis with NMO. But we have also to concede that this is an area where we would hope that we could make much more progress, and we certainly don't have established biomarkers to predict therapeutic response on a soluble level. One aspect which could be related to the work we are doing here, or we are referring to, is that to discover and validate those biomarkers, you probably need very often larger sample sizes and longer-term data than you generally can achieve in the academic setting with the small trials you can do there, where this research has been done. So we believe that we are in general agreement with the draft guidelines concerning soluble biomarkers, but we would think it would be worth at least mentioning that development program could be a potential vehicle in which biomarker work, biomarkers research and validation could be enhanced. Let me now turn to MRI biomarker, and here several has been established for many years, starting with the interferon beta 1b registration some 20 years, or 20 years ago, exactly 20 years ago. And the guideline acknowledges, obviously, the role of those MRI measures to screen anti-inflammatory effects and in exploratory trials, or to monitor CNS lesions or inflammation in longer-lasting pivotal trials. The guideline also mentions that these MRI measures, as we use them on a standard level, may be less useful to study effects on tissue loss or potential drug effects beyond inflammation. And in that context, the guideline suggests that CNS atrophy measures or new MRI techniques may be of potential relevance in the non-relapsing SPMS or PPMS indications. We do, however, feel that that probably is a little bit too limited, and we would ask to consider whether at least whole brain volume loss as an MRI outcome could be better reflected for its potential usefulness in drug development, and exactly in the space if you go to neuroprotection and if you go to areas outside of inflammation. The reason why we believe so is there is, on the one hand, relatively relevant academic support that this may be the most advanced imaging biomarker covering brain tissue preservation. There are well-developed methods around, at least at some specialized sites, to quantify volume change successfully, and this has been done in multi-center clinical trials in several instances. Though we have to concede that there are still methodological differences between the methods used, but nevertheless, within a method, if that is used consistently, that would make sense. In those trials, and this is important for the drug development purpose, obviously, sensitivity to pharmacological interventions in the 1 to 2 year time frame in relapsing MS has been shown, as well as in exploratory trials in SPMS. And there are, from a clinical relevance point of view, modest to moderate correlations to brain atrophy with disability, as well as, again, the work from Maria Pia, correlation of treatment effects on disability progression with treatment effects on brain atrophy. There are potential confounders which could bias results, such as pseudoatrophy or pathologically non-specific volume changes. But academia believes, and also I think the trials which have been conducted and which have been working show that these could be potentially dealt with by either addressing it by the design or by having longer-lasting duration of the trials where these play less of a role. So therefore, we believe that the guidelines should be amended or could be amended to mention whole brain atrophy as an endpoint in exploratory trials, where the objective may be to halt or delay disease ability, and the mode of action is not primarily anti-inflammatory, and to mention whole brain atrophy as a secondary endpoint in pivotal trials, where the primary goal is to halt or delay disease progression. We have also a couple of minor amendments to the more established MRI endpoints. We believe that regarding effects on gadolinium-enhancing lesions in exploratory trials, that this is not only showing anti-inflammatory effects, but could also be facilitating those findings, which already has been done, and I think it could be worth mentioning it, as well as in phase 3 trials, the established outcome measures could be used as an independent and fully blinded source of evaluation and confirmation of drug anti-inflammatory effects. That is implicitly there, but not explicitly stated, and it could be explicitly stated. We touched on surrogacy in the morning, so again, this is quite nice because we would also suggest that the goal to establish MRI surrogacy, as it was already stated in the current valid version of the guideline, should be maintained and not taken out, as it seems to be the case now. I now move to another methodology, and again, I touched briefly upon optical coherence tomography. We have selected this measure because it's probably one of the most dynamically evolving imaging modality outside of MRI, with a high potential to become relevant to measure axonal and neuronal degeneration or loss. And I think we all agree that this is the area where we perceive the gap in imaging or in other modalities to show treatment effects early on, also to help us to do early trials, phase 2 trials, exploratory trials, to see whether a drug may be promising to go further. And that outcome, though still not fully evolved, has some features which potentially lend themselves to a broader use. It's an objective, non-invasive, painless, patient-friendly technology, and it studies in the retina both axonal and cell structures, which seem to be amenable to change over time with ongoing disease, both in multiple sclerosis but also in optic neuritis. And quite nicely, you have a clinical correlate with visual acuity, which you can link to changes you see in the anatomic structures, which is very appealing to many of us, and probably also for regulators at the end of the day, to have that meaningfulness established. The population, again, it's early days in that regard. The population to pick is probably tricky at this time. Certainly for exploratory trials, it may be premature to go directly into general MS population, because the rate of change, as it is currently detected, may be too little to do that in a 1-year study or so. So in a longer term, that may be feasible. So one could consider exploratory trials in acute optic neuritis as a multiple sclerosis proxy, where the changes of the dynamics of RNFL or ganglion cell layer may be in a reasonable time frame. And indeed, there are several optic neuritis trials ongoing, run by industry, and which will show whether this is viable or not, and will go. The main risk here from a feasibility perspective is that these trials, you need an incidence population, you cannot really count how many you will find, and the infrastructure to run those is not well established yet, but that could change over time. So we would assume that it's not prime time for OCT, but we would say that it would be worth probably considering to mention that the improved visualization by and performance of OCT technology could suggest an increasingly important role to measure axonal and neuronal degeneration in the future. And with that, I thank you for your attention.